Hi everyone, I'm Mukaddes Yıldırım from Ankara University, and I'm going to present our research about the effect of PDYN RS910080 polymorphism on the risk of alcohol use disorder in Turkish men. First, I will give some information about alcohol use disorder, dynorphins, and PDYN gene. Then I will talk about our aim, methods to investigate our hypothesis, and finally our results. Alcohol use disorder is a recurrent and complex brain disorder. It's characterized by physiological withdrawal and the emergence of many negative effects during the withdrawal process. AUD is associated with the development of mental and behavioral disorders, a wide range of non-communicable diseases, and traffic accidents. In addition, it causes social and economic problems both individually and nationwide. Although alcohol use is a common behavior, some individuals are more adversely affected by alcohol. One-time use of alcohol causes different results among individuals. This diversity is generally accepted as the result of the genetic differences. Studies on twins, adopted individuals, and people with a family history of alcohol use disorder have shown that alcohol use disorder is related to genetics. Dynorphins are obtained by proteolytic processing of prodynorphins encoded by the PDYN gene and bind to kappa opioid receptors encoded by the OPK1 gene. Molecular changes in the endogenous opioid system induced by alcohol and other addictive substances may cause neuroplastic adaptations that lead to addiction. As a result, dopamine release is blocked and dynorphins have a protective or regulatory role in reducing extracellular dopamine. Substance abuse and tolerance after substance-induced synaptic dopamine increase. High dynorphin levels cause dysphoria and anhedonia associated with negative craving and substance withdrawal. In this picture, you can see dynorphin levels difference between ethanol naive and chronic ethanol exposure neurons. The human PDYN gene contains 4 exons, is located 15.3 kilobase long, and is located chromosome 20. And here you can see our SNP that we studied on. It has been shown that dynorphins encoded by the PDYN gene also cause negative moods. These negative mood states cause negative craving and cause the patient to consume much more alcohol. However, no study investigating the relationship between PDYN gene polymorphisms and alcohol use disorder was found in the Turkish population. Therefore, in this study, we aim to investigate the effect of PDYN RS910080 polymorphism on alcohol use disorder in Turkish men. Although different results have been obtained from genetic studies, PDYN gene variations have been accepted as powerful candidates in terms of alcohol dependence. PDYN RS910080 and PDYN RS2235749 SNPs are both found in 3' UTR of the PDYN gene. It has been shown that there is a high linkage equilibrium between these two SNPs. PDYN RS2235749 has been seen to modify mRNA expression of PDYN gene. The present study was undertaken with the aim of exploring whether PDYN RS910080 gene polymorphism has an effect on AUD due to the contribution of dynorphin to the drug-seeking behavior due to the negative reinforcement. Let's talk about materials and methods. A total of 50 individuals with AUD and 50 healthy controls were enrolled in this study. All AUD patients were entered in the Ankara University Faculty of Medicine, Department of Mental Health and Diseases. They were diagnosed with AUD according to International Classification of Diseases 10 diagnostic criteria and fulfilled the DSM-5 criteria for AUD. We have 3 exclusion criteria for patients with AUD were as follows. Substance use disorders other than alcohol and nicotine dependence, clinically significant comorbid psychiatric illness such as any psychiatric disorders, schizophrenia, mental retardation, bipolar disorder, and severe depression. And the last one is subjects administered drugs for psychiatric illness such as depression. Controls were compromised of healthy individuals that satisfied the same exclusion criteria and had no past or current alcoholism. Controls were matched to AUD patients for gender and smoking habits. Like I said before, a total of 50 individuals with AUD and 50 healthy controls were enrolled in this study. 2 milliliters of venous blood was taken from everyone into tubes with ETA for DNA isolation. Genomic DNA was extracted from whole blood samples using the KIM DNA blood kit. The PDYN gene region containing RS910080 polymorphism was amplified using polymerase chain reaction, and PCR products were digested SDU1 37 degrees Celsius for 1 hour. The undigested and digested PCR products were separated by gel electrophoresis on a 3% agar gel visualized by ethidium bromide staining under an UV illuminator. This is a representative agar gel image of digested and undigested PCR products. On lanes 2, 8 and 9 we can see homozygous wild-type genotype, and lanes 1, 4, 5 and 7 we can see heterozygous genotype AG, and finally we can see 3 and 6 homozygous mutant genotype GG. Our results. The frequencies of the PDYN RS910080 alleles and genotypes were obtained by direct counting, and departure from the Hardy-Weinberg equilibrium was evaluated by the chi-square test. The relationship between the PDYN RS910080 and AUD was modeled by binary logistic regression analysis. P-value was considered as statistically significant. The sociodemographic characteristics of 50 healthy controls and 50 individuals with AUD were presented on Table 1. All subjects in both the controls and AUD patients were smokers. Patients with AUD has a significantly higher duration of smoking and packs of cigarettes smoked in a year than healthy controls. The frequencies of A and G alleles were 69 and 31% in healthy controls and 67 and 33% in individuals with AUD respectively. The genotype frequencies of the PDYN RS910080 polymorphism were consistent in Hardy-Weinberg equilibrium both in patients and controls. The relationship between the PDYN RS910080 genotypes and AUD was examined by binary logistic regression analysis, and the PDYN RS910080 polymorphism was not found to be associated with the AUD. In conclusion, in this study indicated that the PDYN RS910080 polymorphism has no effect on the risk of AUD in Turkish men. A clear generalized statement about the role of the PDYN RS910080 polymorphism can be done with a larger sample including women alcohol users. And these are the references used for research. This is our research team. Thank you for your attention. If you have any question, I will be happy to answer them.